Okay, again, I appreciate the invitation and being able to talk to you about some of the work that we've been doing. And so really the focus of what I'm going to talk about today is really just how you can make sure that you can optimize your methods and also make sure that you have control of your methods and that you really understand what the parameters are that are going to influence the stability of your methods. And so again, we're going to talk about electrospray and the chromatography side of things. And so we'll obviously be in negative mode using C18s, but we'll spend most of our time talking about the compositions of the solutions that we're using here in our mobile phases and what we're going to do to try to maximize our response and maximize our separations. I want to spend at least a little bit of time thinking about this figure right here, and again, what the role of the ion pair is. And so clearly the ion pair helps our spectra, it helps our separation. But I think one of the things that people don't necessarily appreciate is the mechanism of desorption itself and the role that the ion pairing agent plays in the desorption mechanism here as well. And so the ion pair really is here to chaperone your oligo to the surface of an electrospray droplet, and the oligos themselves are going to desorb from the surface of that droplet. And so the ion pair has a really important role in electrospray. And so I think a lot of people who have thought about the ideas of figuring out ways to remove the ion pairs from the mobile phase and perhaps use mixed mode columns that may be able to give you the separation and are worried that the ion pair themselves are hurting your electrospray response. That is absolutely not true. And the ion pairs themselves actually have an important role in the sensitivity of the electrospray method, and it's going to be difficult to actually remove them from the mobile phase without really hurting your methods. Okay, so what are the issues we're going to deal with here? So again, Brian's talked a little bit about some of this, but I've got some other data I want to show you. We'll talk again about the ion pairs, the fluoroalcohols, organic solvents, which is something we haven't really touched on, and my newest area, which is really non-specific adsorption. So again, here's a different view of some of the data that Brian showed you. And so this is just looking at an antisense siRNA strand here. And the idea that we can look at response as a function of the ion pair that we're using. And so at the bottom here, you'll see TMEDA, which is, I'm going to tell you, is always the worst responding ion pair of all time. And so actually we like it because we normalize everything to it because it's always bad there. But again, depending on what strand I'm running, the sequence of responses from these different ion pairs will be different. And so in this particular ion pair, this diisopropylamine is actually my highest responder, and it's about a factor of 5 better than the worst responder that I have here. And so again, it's always important to think about how much you can play with your method sensitivity by which ion pair you select. And for us, it's usually always somewhere between a factor of 3 to a factor of 5 in your method that can be gained by looking at your ion pair. Also, I wanted you to look at the concentration, the way this works here. You'll note that the response goes up as you raise your concentration up. And generally anywhere between, say, 10 and 20 works pretty well. And as soon as you go past 20, you'll see the sensitivity really falls off. And so again, don't take your ion pair above 20 under any circumstances, is what I'm going to say. But again, if you're below 10, you'll gain in method sensitivity just by coming up a little bit in your ion pair concentration as well. So what about the fluoroalcohol? So again, we spent a lot of time early in my group focused all in on the ion pairs. And at one point in time, I finally asked, this is the question. I said, well, what's the fluorinated alcohol doing here? And is there anything else we can use besides HFIP as a fluoroalcohol? And would that gain us anything? And so we looked at a bunch of different fluoroalcohols that were possible to try here just to see what they would do. And so here's looking at TEA as a function of different fluorinated alcohols. And so what you'll note here is that, again, if I didn't put a fluorinated alcohol in here, I'll get a certain level of response. I put HFIP in there, it works a little bit better than not having any fluoroalcohol in there at all. And if I try some of these other fluorinated alcohols, at least for TEA, they're all a little bit worse. Unless I try the NFTB, which is a non-fluoro tributanol, and then that's considerably worse there. Although that's got some fascinating other properties in terms of, if you're interested in higher order structure of oligos, you should look at it. If you're not interested in higher order structures, stay away from it. So if we look at another reasonably polar ion pair like DIEA, again, it's about the same. So again, HFIP works well with the more hydrophilic ion pairs. So the less polar ion pairs like HFIP. But if you go to some of the more hydrophobic ion pairs, like DMCHA here, then you'll essentially double your response if you move to any of the other fluorinated alcohols and move away from HFIP. If you move to one of the, say, really hydrophobic ones like octylamine, again, you can gain as much as a factor of 4, moving off of HFIP to one of the other fluorinated alcohols. And so here's just a simple example here of an LCMS trace. This is a 24-mer, I think, and a 23-mer there, and we're just showing the separation of there. And all this is the exact same ion pair, and I just switched it from HFIP to HIFMIP, so the hexafluoromethyl isopropanol there. And again, you'll note that you more or less go up by about a factor of 2.5 there by making just the switch in the fluoroalcohol. So this is another interesting one that really nobody has done much with this ever since we put this figure out almost a decade ago at this point. And so it's this idea of looking at other solvents. And so there's been a little bit of work with some people that have used acetonitrile. And again, acetonitrile is not necessarily any better or worse than methanol, except for the fact that some of the more hydrophobic ion pairs, you need acetonitrile to solvate them because they'll fall out of solution if you use methanol. So if you really like to move to things like hexylamine and octylamine, you're going to need to get rid of your methanol, and you're going to have to switch to acetonitrile in order to get those to work, but they will work if you do that. But one of my students one day was just doing some flow injection work with isopropanol and came back and said, you know, isopropanol works about 3 times better than methanol or acetonitrile does for a response. And I said, well, that's great, other than the fact we can't use that as an LC solvent because the LC will explode if I put isopropanol in it. But I did say, you know, try, since that's a C3 alcohol and you've already tried the C1 alcohol, I said, why don't we try the one in between those two? And if you switch it to ethanol, it actually works really well too. And so that's actually another way you can essentially get yourself another factor of 2 or 3. And it doesn't really do anything to your chromatography. It'll work just about as well as methanol there as well. All right, so this is my new one that I'm starting to really worry about. And so this is non-specific adsorption. And so we dealt with this for the very first time really early on and talked about it in a paper that are cited down below in 2013. Plus there's another one we've got at 2013 also in J-Chrome-A, where we just talk about non-specific adsorption. And so one of the worst things we had was that early on in sample prep, and again, it's just recognizing the materials that you're using in sample preparation can have some pretty catastrophic effects on how much of your sample you're losing. And so the early one that was really getting us were sample tubes. And so clearly switching off of any other type of sample tubes into the Lovine tubes really helped us quite a bit there. The other one was then our non-specific losses that we were suffering actually to our LC system, although it's taken us a long time to recognize it because I originally attributed most of these losses to sample prep problems, but they're not. They're actually problems with your non-specific adsorption in your LC. And so a lot of this we've tried to deal with by passivation essentially of your system. And so most of us understand this idea that you need to inject a fair number of injections into your system in order to stabilize what's going on. And so what we figured out now is it seems to be that that's maybe 20 micrograms of material that you've got to get into your LC system in order to be able to try to deal with the non-specific adsorptive losses, because that's about what the capacity of the non-specific sites are in your LC. And so again, if you were to take, say, a brand new column out and put it onto the instrument and then make an injection, you'll probably see nothing because you'll lose everything to non-specific adsorption there as well. And so this is essentially this idea of passivation. And so we dealt with this initially by raising the concentration of our internal standards and essentially using high concentrations of internal standards as a way to passivate our systems because we were interested in bioanalysis at that particular time. So certainly for looking at impurities, the idea is that you have a high concentration essentially of your main API, and then that serves to passivate your system as well, since its concentration is also really high. So I think one of the things to really understand here, though, is how do you unpassivate your system? And that's really what we've been spending a lot of time looking at right now. And so what I'll tell you is one of the big roles and we've been working a lot with Martin Galar at Waters looking at this too, but the pH of your mobile phase will matter quite a bit to how well you can keep your system passivated and how rapidly it will unpassivate. And so the lower the pH of your mobile phase is, the faster your system unpassivates. And it can unpassivate in as much as one injection. You can start unpassivating your system. And that means that when you go to make your next injection, so the things that start, once you start separating out in the column, the low concentration early eluders can be non-specifically lost, and you just may never even see them there as well. So you've really got to be able to keep control of system passivation, because I think it's really a significant problem. And so that's, again, one of the things we're working on now, and I hope I'll get most of this data out into the literature, because there's some pretty alarming data that we've got on this. All right, so what about chromatography? So let's switch to chromatography. One of the good things on the chromatographic side is pretty much all the ion pairs, if they will work, will give you pretty similar separations. And so really I think you can select your ion pair to optimize your mass spec response, and you don't necessarily need to worry that much about it really being bad for your chromatographic separations. So again, other than ones like TMEDA, which is never going to work, for this PA one here, which I think is it's penta, it's a 5-carbon alkylamine here, and that one doesn't really work either. So this is another piece of data that was really causing me a lot of concern the first time I got it. In fact, it took me about a year just to wrap my brain around this figure. So again, this was part of our study that we were just doing before, this idea that we would look at a range of concentrations of alkylamines and try to get a sense of how they separated out. And so the student came back to me and said, okay, so I keep stepping up the ion pair concentration, but after I get up above, say, 15 or 20 millimolar, the retention actually starts getting worse and worse and worse and worse and worse, and eventually just completely goes into the void volume. And I said, well, that's a serious problem, because this is supposed to be ion pair chromatography, and that's not the way ion pair chromatography works. Ion pair chromatography works by increasing the ion pair, and we get more retention up until the point at which, at some point in time, maybe your counter ion starts repulsing it, and you'll get a little bit of rollover at really high concentration, but not like goes into your void volume problems here. And we tried it for a DNA, we tried it for a modified DNA, so this is a phosphorothioate DNA, and it's exactly the same. And we tried it with every different alkylamine we have, and they all do this. So I kept thinking and thinking and thinking. I said, what in the world is going on here? And so at some point in time, we finally said, all right, let's do a light scattering experiment on the mobile phase. And so we did that, and we found that there was significant scattering going on from just the mobile phase. And so we decided to just take a shot at it and take it up to get a transmission electron micrograph of the mobile phase itself, just so we could get a visual on what's going on here. And you'll see these little aggregates or these colloids in the mobile phase. And we found that we can control this for a number of different ways. I can control it with concentration. You could actually control it with temperature, which goes back to another point that Brian made earlier, this idea that we like to run at high temperatures. And it turns out actually the higher you make your column temperature, the more of the colloids you're going to form in your mobile phase. So these are actually pretty big and happen at a pretty, like I said, you can see on the one on the left, it's pretty much you filled your entire mobile phase up with colloids. So what are these colloids really all about? And so the idea here is that you're going to have a mechanistic switch that explains this loss in retention. And so you're going to reach a point where on the left side here, you're going to see typical ion pair chromatography. And so this is the way it's supposed to work. Again, the ion pair sort of embeds in the stationary phase and gives us this charge on the surface. That's going to be our main retention mechanism, and that's the way we want ion pair to work. So what the colloids end up being essentially are micelles of your alkylamine that may or may not have the fluoroalcohol in there as well. I'm not sure yet whether what the role of the fluoroalcohol is in this, although I do think it is involved in the colloid formation as well. And so I think that's a lot of why when people like 20 or 25 years ago did some of those concentration experiments with TEA acetate, you don't see this impact, but if you do this experiment with TEA HFIP, you do see this effect. So the fluoroalcohol must have a role in this as well. So again, you'll form these micelles, and so you get this micelle up in the mobile phase, and so essentially your oligo becomes attracted to the micelle and doesn't end up being attracted to your stationary phase, and so your retention goes way down. And so I think one of the big issues with this is that this micelle formation has a kinetic component to it, and that this is what everybody refers to as mobile phase aging, right? So that you make your mobile phase, and your mobile phase works really well today, maybe works well tomorrow, and then it stops working so well. And this is the problem that you're having here, is that your mobile phase is making these colloids in there, and the colloids are impacting the reproducibility of your chromatography. And so the higher the concentration, the faster it happens, higher the temperature, the faster it happens. But again, there's going to be a time component to it. If you let any mobile phase sit around long enough, it's going to do this. So we did recognize that pH, and this is what I'm working on right now, is to try to figure out exactly where this is, but there is definitely a pH impact to this as well. So again, as you run the ion pair concentration up, you're raising the mobile phase pH as well. And so once you get the pH up, then at a certain level, then again, these colloids will start to form, and then your retention will crash down, say around, say, 50 millimolar as well. But if you can stabilize the pH in there, you can change the concentration of the ion pair, and then the red line here is exactly what you expect ion pair chromatography to look like. Again, that you raise the ion pair concentration up, and then it kind of levels off, and then it gets high enough, you'll start to see it roll over a little bit at really high concentration. So that's exactly what ion pair is supposed to look like. And if you will control the pH of your mobile phase and don't let it keep changing on you, then you actually can get ion pair behavior across the high range. And so what this all is coming down to is that there is sort of a pH that you need to stay under in order to keep this. It's probably going to be a little bit different for every ion pair, since they all have slightly different pKa's. Though they're all relatively similar, but there's about a half a pKa unit range to the pKa's of all the different ion pairs. And it looks like it's going to be somewhere around 9. There, maybe a little higher than that, but not a whole lot higher than that. So again, and so that may be part of this as well. Again, that if you don't know what the pH of your mobile phase is, you absolutely do not have control of your mobile phase and your chromatography at all. All right, so where are our challenges here? So again, I'm going to just start with this idea that there probably is not a universal mobile phase, unless you don't necessarily care about optimizing your sensitivity, and then because there are lots of mobile phases that are going to work, it's just that you can absolutely tune your mobile phase in for your specific oligo. There is definitely still a lack of understanding of the retention mechanisms that are in play here. And for impurity and degradation product analysis, again, control of adsorption is going to be a big part of this, and the LC system passivation is a serious problem here. Another issue we've had a little over the time is just controlling adduction and also really controlling oxidation. And so we've had some issues where we've found some conditions where I can oxidize the samples during sample preparation, and they're not actually any part of the samples themselves. So again, just to give you a chromatographic sort of look at what we have here. So again, this is just looking at a oligo that we had ordered from a commercial vendor here that they sent us to us. And we found lots more impurities than the vendor thought were in it. But again, I would agree with the earlier idea that, again, using a combination of LC-UV and LCMS is probably really the way to go here to be able to look at both the early eluting, all the things that are co-eluting in the main peak, and then to be able to look at the late eluders. I'm just going to throw out two quick things for you. Again, I think one of the bigger challenges we have right now is going to be methylcytidine degradation, and Ionis has done some really good work on this early on, although it has not gotten down much below about 1% for this. And so we've got like about this one of the larger FTMSs in the US right now, and I'm getting ready to try to see if we can really get this down to the 0.1% level in order to be able to characterize that. So I will get back to you on that there. And then the other thing is, again, what happens to your oligo when you do sterilization of it prior to release? And in particular, the methylcytidines, I suspect, are the biggest problem you're going to have when you sterilize these things. I think you're going to lose methylcytidines, more methylcytosines from that.